If we look here at the overall process of DNA replication, as we move down and we look at these different stages, notice that the replication bubble is growing. And the replication bubble is growing because the helicase enzyme is breaking through these hydrogen bonds in both directions. And it's going to continue to do this as we synthesize more and more of the new DNA that we want to make. So we're going to continuously open up this replication bubble, and eventually we'll run into the nearby bubbles because this is really just a section of a chromosome that we're copying. Now, if we're making our new DNA and we just draw it here at the beginning, here would be a new piece of DNA, and here's another new piece of DNA, which we would be adding onto a primer that would be there. So I could draw in my little piece of primer if we wanted to. So short piece of primer, we extend it with the DNA polymerase. And then I'm just going to draw a little arrow here on my 3' end of the new DNA because the 3' end, which again is my arrow end, is growing all the time. So we're extending that. And remember that we can't really add on to the back side of the primer because DNA polymerase is not going to be capable of extending a 5' end. So what that means for us is that we will have essentially 2 different strands to our new DNA with respect to a replication fork. So if you just think about dividing all this right down the middle, and we're just going to talk about one side at a time. So if we just focus over here on this side, notice that this top strand that we're making, and so by top strand I'm talking about this right here, that is going to be what we call the leading strand. And by leading strand we're referring to the new DNA that we're making. The reason we're going to call that the leading strand is because the 3' end of the new DNA points to the fork. And when you talk about what that ultimately means for the synthesis process is that we only need 1 primer, and once we have that 1 primer, we can just keep extending that end as the helicase moves further and further. So if we draw that in the next picture, I've got my primer there, and my DNA polymerase is just going to keep making this green even longer and longer. And we can look at that in the bottom picture here as well. And again, it's one nice continuous piece until we run into the next strand. And if we looked at the other side of my diagram, the other replication fork, we would see essentially the same thing. We need only 1 primer, and once we have that 1 primer, we can just kind of continuously extend it because the 3' end is pointing in that direction. So on the other side, the bottom here, this would be my leading strand for the new DNA. On the other end, we have a little bit of a problem because we are going to have to keep backing up and adding a little primer and extending that. So we will do that, and eventually it runs into the previous one, and we'll end up removing that. But once the helicase moves again, if we just kind of show where this is all going to end up, notice that as the helicase keeps moving, we have to keep adding a new primer. So primase is going to have to keep coming in and adding a primer as we continue this whole process. And so we will end up with lots of little pieces or fragments of DNA. And this again is because of just the directional synthesis of DNA polymerase, the fact that it can only add to a 3' end. So when we look at this other side, so down here it's going to be shown in red. Okay, that is what we refer to, and I'll just write it over here, that's what we refer to as the lagging strand. The lagging strand, if we want to just identify it, notice that the 5' end of the new DNA points to the fork. And ultimately what this means is that we need lots of primer. We'll have to continuously add new primers as the fork continues to move back, and we'll get fragments of DNA. which we will want to join together later on to get that continuous piece of DNA. So don't forget that eventually we will be removing these primers. The DNA polymerase is going to really be kind of running right over the top of them. And then we will need that DNA ligase to seal the gaps. So we will need lots of ligase. All of these steps here, they're going to make that strand take a lot longer to synthesize, hence the name lagging strand. Its synthesis lags, or it's slower than the leading strand is going to be. Notice that these terms, the leading strand and the lagging strand, they do refer to a particular replication fork. So in other words, if we look at this top picture, notice that the leading strand, if we're talking about the replication fork on this side, it's the top strand. But if we talk about the leading strand with respect to this replication fork, it would actually be the bottom strand. And the lagging strands, if I just went through here and I showed them all in red, here are all of my lagging strands. Now these lagging strands, we said they have lots of little short fragments of DNA on them. We do have a name for those little fragments, and that's they're named after the individual that discovered them, but they are called Okazaki fragments. Okay, so those are just the short little pieces of DNA that we do get initially when we're synthesizing this lagging strand, and ultimately at the end we are going to go and we're going to join those together. Just a few last participants in this process that we do want to mention. We do have one protein that's going to be called a sliding clamp, and that is really exactly what it does. And so the sliding clamp is going to assist DNA polymerase and prevent it from falling off the DNA or falling off the template. So we will see this located right there next to DNA polymerase. So my DNA polymerase is going to be located right there, and we're going to have another one in this picture located over here, kind of working. And so the sliding clamp is just moving along with the DNA polymerase, again, helping to ensure that it does not come off of the template until really we're ready for it to come off the template. The other final enzyme, that enzyme's name is going to be topoisomerase. And topoisomerase is going to be located back here where the strands are still connected together. And we also think of it as being kind of a donut that encircles the DNA strands. And so I'm drawing it on both sides there. Keep in mind that my DNA sequence would continue here. So it is around the DNA that's still double-stranded, and we would call it behind the replication fork. And what topoisomerase does is it's going to fix what we call over-twisting or over-winding of the DNA strands. And the reason we get this is because this is a helical molecule, and the helicase that we have in there, shown in blue, is really kind of pulling these two strands apart. So you may have noticed this before if you take something like a piece of yarn and you try to just grab hold of a couple of the smaller strands that make it up in the middle and you start to pull them apart, you get kind of this over twisting or this knotting that starts to occur a little bit. And so that's the same thing that's happening here with our DNA molecule, and topoisomerase is going to help us to fix that. So what topoisomerase is going to do this is it's actually going to cut one of these two strands, and when it cuts that, it will allow that strand to kind of unravel or untwist itself. And so once it has kind of spun around and untwisted itself, then topoisomerase is going to put it right back together. And it would do the same thing on the other side. And we're not calling this a sliding clamp because the sliding clamp is the one that specifically assists the DNA polymerase, but it does essentially just continue to slide in this direction as the helicase enzyme continues to work. And whenever we need it to correct the over-twisting, it will correct that over-twisting for us.